The purpose of this video is to explain the process of calculating CFUs per mil. The process of calculating CFUs per mil, or to count the number of organisms in a sample, is dependent, or it can be obtained by two mechanisms, two procedures. So this is quantitating bacterial numbers, and we can do these directly through a direct method, which entails diluting a sample and then plating that sample. By this method, we can directly count the number of cells in the desired sample. So this is a direct method in which the actual cells in the sample plated are being counted. We're counting what are called viable cells, meaning cells that are alive. The plating aspect of the technique can be obtained by a pour plate, spread plate. A pour plate is a technique in which the volume of sample that will be plated is mixed with melted and cooled agar and then pour onto an empty sterile plate. This allows for the growth of the bacteria inside the agar and on top of the agar. The cells inside the agar will create small colonies because they're constricted by the agar. The cells on top of the agar will have larger colonies because they're not constricted by the agar. On the other hand, in the spread plate, we're going to be spreading the sample on top of the plate. All the colonies will grow on top of the plate, and they will be counted, and that will be accessible. So either technique would work, with the caveat that the pour plate gives us more area to spread our cells. The idea here is that each of the cells that fall on the agar are going to create one colony. So by counting the colonies, or CFUs, you're essentially counting the number of viable live cells and the volume of the diluted sample that was plated. Once you know how many cells in the volume of the diluted sample plated, all you need to do is multiply by the dilution factor, account for the volume plated, and you have your CFUs per mil. So the first part of this technique is to create tenfold serial dilutions of the sample. And the reason why tenfold dilutions are made usually is because 10 and multiples of 10 are easy numbers to multiply and work with. So that's why the number 10 is used. So we're going to dilute by factors of 1 to 10, or 1 to 100, or 1 to 1,000, because it is easy to multiply 1 to 10 times 1 to 100 times 1 to 1,000. So we have our sample that we're going to need to count. The sample is in a broth, so we can make the dilutions. We're going to create 3 serial dilutions, and we need to figure out the CFUs per mil in the sample. So the first step is to create the dilutions. We're going to make 3 serial dilutions, and for the sake of this explanation, we're going to make each of them different. They're all going to be tenfold, but they're going to be used in different numbers. So this is not exactly the same numbers that you need to work with in your lab report. So first we need to add the diluent, the water, onto each of the tubes. This is the fluid that will be used to dilute the bacteria. So in the first tube, we're going to add 9 mils. In the next tube, we're going to add 0.9 mils. On the next tube, we're going to add 99 mils, sorry. Okay. So 99 mils is the biggest volume, so let's make that volume bigger. 9 mils is next, and we'll leave the 0.9 as a small volume, which is what it is. So notice we don't have to add the same volume to each tube, but it's typically helpful if we do. Again, we're adding different volumes so that we can practice with different kinds of numbers. Dilution factors, the formula is the volume of the microorganism over the total volume. But keep in mind the total volume involves the volume of the microorganism plus the volume of the water. Okay. So, we're going to proceed to do the dilution. 1 mil of the original sample microorganism is going to be added to the first tube. Then 0.1 mil will be transferred from tube 1 to tube 2. And then again, 1 mil will be transferred from tube 2 to tube 3. Okay. For tubes 2 and 3, as we calculate the dilution factors, we're going to need to multiply by previous dilutions. For tube 1, there are no previous dilutions, so we don't have to account for previous dilutions. But when we calculate dilution factors for tubes 2, we have to account for the previous dilution, and the same thing for tube 3, we have to account for the previous dilution. Okay, so we're going to recreate these dilutions that we've made. Tubes 1, 2, and 3. Remember that tube 1 had 9 mils of water. Tube 2 had 0.9 mils of water. Tube 3 had 99 mils of water. And then we transferred 1 mil of the original sample into tube 1, 0.1 mil of tube 1 of dilution 1 into tube 2, and 1 mil of dilution 2 into tube 3. Okay, so let's calculate dilution factors for tube 1. Volume of sample of microorganism over total volume. All right, so the volume of the microorganism that was placed on tube 1 is 1 mil. Total volume of the sample will be the volume of the water plus the volume of the microorganism, which will be 10 mils. Mils are going to cancel, the units cancel. Dilution factor should not have units. That gives you a dilution factor of 1 to 10 for tube number 1. So tube number 1 is a 1 to 10 dilution of the original sample. Now let's calculate the dilution factor for tube number 2. Volume of microorganism is 0.1 mil. Total volume, 0.1 mil plus 0.9 mils. The units cancel. And remember, because this is the second dilution, we are going to have to multiply by the previous dilution. So now we have 0.1 over 1 times the previous dilution, which is the dilution factor for tube 1, which is 1 to 10. So when you do the math, dilution factors for tube number 2 are going to be 0.1 over 10. However, it is incorrect to have a decimal in a fraction. So 0.1 is a tenth of a number, a tenth of 1. We need to get rid of the decimal. And so we can multiply numerator and denominator for a number that will allow us to get rid of that decimal. Because 0.1 is a tenth, we can multiply it by 10, and that will get rid of the decimal. But if we multiply the numerator by 10, we also have to multiply the denominator by 10. So multiplying numerator and denominator by 10 will give us 1 over 100. So the dilution factor of tube 2 is 1 to 100. Tube 2 is a 1 to 100 dilution of the original sample. For tube 3, we're going to try to fit those calculations on the corner so we don't have to erase everything and start over again. So dilution factor for tube 3, volume of the sample is 1 mil. Total volume is 99 mils plus 1 mil, which gives us 100 mils. Remember that the units are going to cancel. But also remember, we have to multiply by the previous dilution, which is the dilution of tube 2, which is 1 to 100. So finishing the math, dilution factor 3 is a 1 in 10,000 dilution. So tube 3 is a 1 to 10,000 dilution of our original microorganism. 1 to 10,000 can also be expressed as 1 to 10 over 10 to the 4th, which is easier to write than 1 to 10,000. Okay, so now we have our dilutions. We know the dilution factor for each of the tubes. What we have to do next is calculate CFUs. So what we're going to do is we're going to take a sample of each one of those dilutions and plate it. So I'm going to recreate the dilution, the serial dilution. And again, we're just going to plate a volume of each of the dilutions onto a pour plate. We're going to dilute in a pour plate. Okay, so just very quickly, tube 1, tube 2, tube 3. Tube 1 was a 1 to 10 dilution of the original sample. Tube 2 was a 1 to 100 dilution of the original sample. Tube 3 was 1 to 10,000 dilution of the original sample. Now the volumes plated don't have to be the same. So again, for the purpose of showing how this process is done using different volumes, we're going to go ahead and use a different volume each of the samples. We're going to plate using different volumes. I understand that in the lab report, the same volume is being used just to simplify things, but you should understand how this works regardless of what volume is being used. So let's say that in tube number 1, we're going to use a 0.1 mil sample of the tube and that on a plate. Now, tube number 1 is the highest, is the concentration. So it would make sense to try to use a small volume. That way, we have fewer cells, and hopefully we'll get a good plate. Tube number 2 is more diluted. So we'll go ahead and still use a small volume nonetheless, because it's still, somewhat concentrated. So that small volume represents a tenth of what's in the tube. So we have to account for the fact we're only counting a tenth of what's in the tube as far as number of cells. So in our calculations, we definitely have to account for the volume plated. Now, tube number 3 is very diluted. So we're just going to put a 1 mil in the hopes that that will give us enough cells to where we can count. That's a consideration when choosing a volume to plate, is how diluted is the sample. If the sample is very diluted, you might want a big volume, otherwise you may not end up with enough cells. The formula to calculate CFUs per mil is the number of colonies that are going to grow in the plate over the volume plated times the dilution factor of the sample plated. Now this is the deal. CFUs per mil of the original sample, that's what we're going to try to calculate. So if we calculate CFUs per mil of plate 1, 2, or 3, the numbers that we end up with should be the same, because what we're trying to know is how many organisms per mil in the original sample. And that should not change whether we use plate 1, plate 2, or plate 3. So ultimately, you don't have to calculate CFUs for each plate. Choose one plate, whichever one has the best numbers, and that should be all you need. Now, the number of organisms in a plate may change depending on the dilution sample. So tube 1 was very concentrated, and even though we put a small volume, you may still have so many cells that you will not get isolated colonies. You may get confluent growth, where it will be very difficult to count every single colony because they're so close together and so many of them, there's no way to count them. On the other hand, tube 3, plate 3 from tube 3, dilution 3, may have very few colonies, maybe 1 or 2, which may be statistically insignificant, whereas that's not a good number. So there's typically a range of number of colonies that is considered statistically significant. And we take the 30 to 300 range as the range that we typically use. Now the range may change depending on the procedure that you follow, but in our case, we're going to use the 30 to 300 range. So any plate that has less than 30 colonies is not a good plate. Any plate that has more than 300, it is deemed those are too many colonies. That would be too hard to count. You could make mistakes. It's not a good plate. So we want to choose a plate that has between 30 and 300 colonies. You could have 2 plates that fit that bill. Either plate will be fine to calculate CFUs per mil. Again, to calculate CFUs per mil using plate 1, 2, and 3, you have 3 calculations. All 3 numbers should be very similar. All right, so let's say plate 1 had tons of colonies, so many that it's just, you can't count it. So you report that as 2 numbers to count, or TNTC. 2 numbers to count. Not a good plate. So let's see what happened with plate number 2. Let's say that plate number 2 ended up giving you 280 colonies. And when you count the colonies, you count both the colonies on the surface and the colonies inside the agar, which are tiny colonies. So you look for those tiny colonies because you need to count every single colony. The colonies that you're counting represent the number of cells in the volume plated, which in 0.1 mil, which in this case, in the case of plate 2, is 0.1 mils. So 0.1 mils of dilution 2 had 280 organisms because it gave you 280 colonies. Okay, that's a good number. It's between 30 and 300, so you could use that plate. Let's say plate number 3 had 30 colonies. Again, that's within the range, so that's a good number also. So you can use either plate to calculate CFUs per mil. Both plates will give you a valid number. Both plates should give you a number that is very similar. So let's go ahead and calculate CFUs for plate 2. We're going to go ahead and calculate for both plates, and that way you'll see that both plates give you good numbers and give you similar numbers. Okay. CFUs per mil, number of colonies 280, volume plated 0.1 mil, times the dilution factor, which is 1 to 100. That's the dilution factor for tube number 2. You do the math, 280 CFUs over 0.1 is essentially a 1/10. So you're multiplying 1/10 over 1 to 100, which gives you 1 to 1,000. Now, this is mils, don't forget the units. So what you're looking at here is essentially the division of 2 fractions. So you're dividing a fraction 280 CFUs over 1 divided by the fraction 1 mil over 1,000. So you essentially multiply the denominator of the second fraction against the numerator of the first fraction. So 280 CFUs times 1,000, and that gives you the answer, which is 280,000 CFUs per mil. If you express it in scientific notation, which is what you should do, it will be 2.8 times 10 to the 5th CFUs per mil. So that's how many colonies, how many cells, I should say, per mil in the original sample, 280,000. Okay, now that should be enough. That's all you need to do. You've calculated the CFUs per mil. However, just for the sake of this explanation, we're going to go ahead and calculate CFUs for plate 3, and we should end up with a similar number. So if you use the values for plate 3, we have 30 colonies. Volume plated is 1 mil times the dilution factor, which is 10 to the 4th, and that will give us 30 CFUs over times 10 to the 4th, which is essentially, so that would be, let me go back here, 30 times 10 to the 4th CFUs per mil, which in citation will be 3.0 times 10 to the 5th CFU per mil. So what you're getting here is 280,000 CFUs per mil with plate 2, 300,000 CFUs per mil with plate 3. 20,000 difference is really not anything to be concerned about when you're talking about these kinds of numbers. So this is essentially the same number. The difference accounts for maybe mistakes in dilution. Some cells are left behind. Some cells may be killed in the process. So those are the kinds of factors that does not give that account for the differences. Mostly human error. Mostly differences in pipetting. So that is essentially, those two numbers are essentially the same. Either one you report will be correct. Okay. So hopefully this helps. Expect this kind of problem in the practical.